Welcome to Ask the Expert. This is a brief, informative conversation where we talk about your work and invite the audience to ask questions and participate in the conversation. I am hoping you can just give us a little bit of backstory about your career and what led you to the Melton Lab, and what's going on with you right now. I'm going to let you sort of like fill in those blanks.
Okay, so well, thank you for having me, first of all, and good morning.
Good morning.
So I'm Elad Sintov. I'm former postdoc in the Melton Lab. So I started this career path in my PhD, where I did worked on diabetes research, mainly on primary human islets, where we worked on to understand the differentiation that happens in natural pathogenesis of diabetes, but also looking at a model in vitro where we could see the differentiation, but also find ways to reverse it and induce redifferentiation of beta cells after expansion in vitro. Naturally, I wanted to continue that path with diabetes, specifically type 1 diabetes. I moved with my family to Boston and joined Doug Melton's lab as a postdoc to work mainly of everything that involves immunogenicity of stem cell-derived islets or beta cells, either in an autologous autoimmune model setting, or what I'm going to show you today, some work in recent years about everything involving alloimmunity of immune rejection of stem cell-derived islet that we anticipate to happen when we transplant those cells in diabetes.
That sounds great. And so in some ways, this is your quest to find, the invisibility cloak, right?
Yes. So finding targets to address the fact that they're prone to be immune rejected. So finding gene editing targets where we can reduce that or avoid that. And as you said, making them invisible to the immune system.
That sounds good. Okay, take it away.
Okay, so if this is Ask the Expert, you can ask me, but I was mostly surrounded by experts in the Melton Lab. In other labs, I was surrounded by great scientists, starting from our A levels to postdoc and grad students. And it was a great time to learn and to be influenced and to be exposed to this kind of high level of creative thinking. And I have also thank the founders of this project, not just to the lab, especially JDRF, who funded me personally in the fellowship.
Elad, can I ask you a quick question? Now that
Sure.
Doug Melton has left his laboratory to go to work at Vertex, what happens to the laboratory?
So the lab is still there. These people that you see in the picture are the people of Doug doesn't like it when I say it, so the last generation of the Melton Lab members. Most of them are still there working and to finish their projects. And maybe within a year, the lab will close. We don't know, but that's more or less what might happen in the next year. But Doug left, but he's still involved.
Well, that's good that people can finish their work.
Exactly. Yeah. So as you heard before from other lab members and even Doug himself, our lab, Melton Lab, and many other labs at companies have worked and working on these large stepwise protocol to generate stem cell-derived beta cells or islet cells. We call them SC-islets. It's stage 6 after they overgone that differentiation, but it's not only beta cells, there's other cell types in those clusters, but eventually you can also enriched in some level to make a more pure population of beta cells.
So even though you've got a pure population of beta cells, do you have some delta, alpha, et cetera, cells sort of in the mix?
That's before purification. So
Right.
most times in this protocol, at least, you get between 20 to sometimes 40% beta cells among other endocrine cells and non-endocrine cells, or cells that did not differentiate well. So in order to increase the level, prepared more to something that is clinical grade, you can basically purify it by using magnetic sorting or some kind of, so these cells can be transplanted, and when transplanted, they were showed to reverse diabetes in animal, and they're starting to show that in humans. But the major concern that we have is that once transplanted, they're prone to be rejected because their source is allogeneic, so another human. So that patient will reject those cells. But also, there's a risk of recurring autoimmunity because the patients have their immune system is primed against beta cells. So we have to address these two problems, and there's ways to protect them either by physical barriers or immunosuppressive drugs, like what's being done these days in clinical trials, or by altering the immune system. One example is using T regulatory cells. But our goal was basically to generate gene-modified lines that can evade the immune system and induce tolerance. So this is one way to show it of what I'm going to present to you today, where we test and evaluate what's the nature of immunogenicity of stem cell-derived islets. So in simple words, we make SC-islets meet an allogeneic source of human immune cells, or PBMCs, peripheral blood mononuclear cells, which can be easily isolated from blood donations. And we put them together in two different models, either in vitro while co-culturing them, or in a mouse that is immunocompromised, but it's made to be humanized by injecting it with PBMCs and make kind of a mouse in vivo test tubes to make them meet together. And we generate some high throughput readouts either by single cell transcriptomic or by doing pool CRISPR screen, which I will more focus here today in our timeline. So the first question is, what's the nature of immunogenicity? What causes this SC-islets destruction that will happen when it's transplanted? So I worked on these two models, where one, the in vivo one, is by transplanting SC-islets into these immunocompromised NSG mice, waiting for some insulin secretion from the graft that can be detected by ELISA in the mouse serum. And then some of the mice, half of the cohort, we inject with human PBMCs. We call it for short HPI mouse, and we compare it to the control without injection, but they are also transplanted with SC-islets. And then we monitor within a few weeks, you would see graft failure in the HPI mouse in terms of human insulin levels that is reduced. And then we go and harvest those grafts from they're transplanted under the kidney capsule. We harvest the residual tissue that is left in the HPI, compare it to control, and that's compared in single cell sequencing.
So just to clarify, the human PBMC are coming from those who have type 1 diabetes?
So no, this is a completely allogeneic model. So
so completely.
So it's just healthy donors or non-diabetic donors, for instance. Yeah, so we only concentrate on what will happen because this is a different human.
Okay.
And then the problem with this model, there's many problems with this model, but one point that I had to address was that looking at this time point was a very late event. So, and looking at early events in this model was a bit tricky. So I went to a simple complementary model where you just put SC-islets in the same dish with the allergenic PBMCs and look within 24 or 48 hours what happens and compare it to SC-islets that were not co-cultured with PBMC. So I don't have a lot of time to show these results, but I can say that enriched pathway included mostly JAK-STAT signaling pathway, which is what is involved in the interferon gamma response.
Right.
Which leads to what I like to call alarm signaling that alerts the immune system and activates T cell responses, but also other immune cell responses. Other genes that come up are apoptotic signaling. That's it makes sense because these cells are being killed over time. And you can also, if I showed you images, you would see that this graft is reduced by size. And what was interesting also that there was genes that showed inflammatory mediated by chemokines and cytokines, and particular one chemokine was standing out, which was CXCL10. And this is an illustration of the pathway where interferon sends that signal to phosphorylate STAT1 that internalizes into the nucleus and orchestrates that interferon gamma genes. Among them are those MHC class 1 that present antigens to the immune system, but there's also inhibitory signals and these chemokines. There's also other repressors, negative feedback response like SOX1. And then concluding all that, we wanted to know what can be done to reverse this immunogenicity response or the destruction of SC-islets, what can survive, what perturbation we can induce by gene editing to make the beta cells survive more. So we took this unbiased approach where we look at all perturbation possible or all knockouts in the cells by whole genome pool CRISPR screen, where we have a pool of lentivirus vector. Every vector carries a guide RNA and a Cas9. And overall, it's a pool of 76,000 guide RNAs, which targets around 90,000 human genes. So every gene is represented by 4 guide RNA. And once transduced into these SC-islets, now we have every cell with a different knockout, but when we are talking about tens of millions of cells that we can produce in the lab, we have replicates of every genes of between 500 and 1,000 replicates of every gene knockout. Now we can
work.
Yeah, and so we kind of have the advantages of having so many cells, and we can do this CRISPR screen. And we transplanted these cells in the same model as I showed you before, in the HPI model, where we transplant in under the kidney capsule and then inject with PBMCs. Wait for the graft to show some failure by lowering insulin level insulin secretion levels, and then extract both grafts that have failed and other grafts that were not injected with mice that were not injected with PBMCs as control. We isolate genomic DNA from those grafts and sequence those barcode regions of the guide RNA, and then look at what's enriched in HPI grafts versus what's enriched in control grafts, and look for perturbation that are tolerizing and enables cells to maintain in the graft and not die. So they're kind of tolerizing genes. And these are one way to show the results, where we looked at an enriched knockout genes in the HPI. And you can really fast recognize again that these genes are part of that JAK-STAT pathway, where we have HLA genes that are activatory to the immune system as they present antigens. You have STAT and JAK, which is part of the signaling mechanism. And then you have one of the top hits is CXCL10, a chemokine that's once secreted and it's induced by interferon, alerts the immune system. And it's one of the initial inflammatory response as we see it, and activates immune cells or recruits immune cells or both. And by looking at genes, now we can know what can be done, what knockouts are beneficial to improve survival.
What's that? What's the deal with the HNR NPA? What is that at the very top there? Sort of outlier 1p48.
Yeah, so this is one of the genes that we still don't know about. It's it's a splicing
variant
component. Yeah. Yeah, so something involved in splicing. So I marked only known genes in the top hit, but there's so many hits here that are yet to be discovered and to understand what's their meaning. So there's a lot to work with these results.
Yeah, it's a very sort of rich environment to start playing around with.
Yeah, and for my comfort zone, I chose genes that are familiar and I know, because beta-2M is known. When you knock it out, it's known to protect any cells, mainly from immune rejection.
Interesting.
So also HLA. The CXCL10 was something somewhat familiar and somewhat unfamiliar in this sense.
Can I ask you just a couple of questions? I was at another meeting and I joined a little bit late.
Hi, Decio.
How are you? How many hours after injection of the PBMCs do you check?
Oh, so it's not hours, it's weeks. In that humanized mouse model, it takes weeks to see some graft failure. It's not very efficient. It's not as we would expect to see in humans if we transplant them.
And you don't get any general effect of the cells on the mouse? It's specific for the graft?
The PBMCs? Yeah, so I didn't mention it maybe. The mice that were used were MHC null. So that can, when they don't have that MHC, their mouse MHC, you can prolong the time that you keep PBMCs in the mouse without any effects of graft versus host.
And the other technical question, so you are using for, let's say, blocking strategies against each gene, but how do you check if the genes have indeed been inhibited?
So basically, you can look, before you do the screen, you can take one guide or several guides for one gene that you know. And for instance, I used a lot of experimentation with this beta-2M guide because we know it has an effect and it's easy to check if beta-2M is knocked out or not. And I used the same vector as the library, which is the lenti CRISPR version 2, where we put the same titer, looked at what's the efficiency and how much of the cells are reducing the level of HLA.
Okay.
Yeah, and basically you get many hits, but not all of them. There's always problem and confidence. So what you always need, if you are interested in one hit, you need to validate it like everything else.
Yeah. And another question, well, first of all, this is a fantastic experiment, very, very nice. Yeah, fair meod, as I would say.
Thank you to that.
when you are collecting the grafts for the analysis, can you do histology in parallel? Because, for instance, CXCL10 is indeed a very interesting candidate.
Yeah.
And T cells that are homing, for instance, they, at least in human type 1, they express CXCR3, which is the receptor for CXCL10. So if you could have histology in parallel,
Yeah.
I don't know if it's technically feasible, you could show if, for instance, CXCL10 is one of the top, that you have more infiltration, and then when you knock down, this decreases. So
Yeah.
this would be an additional proof.
And that has been shown, actually. I think there's one paper from Bartrope 10
Yes.
years ago.
Yeah, this is what I was thinking about.
Yeah. So for our instances, for this part, it's not like they're not like in the pancreas where you have islets and you can look at islets that are infiltrated versus islets that are not infiltrated. We have one graph. I have a picture of that graft being infiltrated by T cells. It's a good point to also stain for CXCR3 for the receptor.
Yeah.
But the problem with my experiment is that I took everything in the late stage, where I didn't also have time to mention that, but in the late stage, there's not a lot of CXCL10. So we think that CXCL10 was selected out first, but we do see it in that first early event and it expressed. And even when we take this late event graft failures or rejected graft, we see that one of the knockouts that were highly enriched is CXCL10, even if it's not expressed there anymore. Meaning in the first few weeks or days, we don't really know yet, when CXCL10 has a big part in setting the fate of the cells that express it. And those that express it might be selected out first. Yeah, so that's where we see it. In the next slide, I can show and we'll show also CXCL staining in the SC-islets, at least in co-culture. If I can also, so there's another part of this CRISPR screen where we looked in the other way. So there's those knockouts that are depleted in HPI or enriched in the control cells. And these depletions basically are depletions of those cells that were depleted with those genes, basically lost something that is essential for their survival under this inflammatory challenge. And one good example is the CD274, which is PD-L1. PD-L1 we know is essential, and it's a good ligand to express and deliver that inhibitory signal. And the fact that we see it here, it's encouraging us to somewhat believe the other gene knockouts, like SOX1, which is a repressor of JAK-STAT. Then there's other things like PTP genes and SUMO1, which is an inhibitor of STAT1 phosphorylation. So potentially we can express or activate these genes to attain immune protection, or we can knock out these genes to get the same goal. So we have a lot of, if we were to go over all this data, we have a lot of work for validation. But with my time frame, I concentrated on especially CXCL10, as we will see, and during co-culture, it does upregulate, same as beta-2M and STAT1, which is the master regulator of the interferon response. And CXCL10 is also secreted over time in these culture with PPMCs, where in those SC-islets that you can, that were co-culture with PPMCs, you see high levels of HLA proteins being expressed. Some cells express CXCL10, those cells that are, I believe, they're a portion of the cells that are responsible, those alarm SC-islets. they have staining for CXCL10. And also these cells have staining for phosphorylated STAT1, which internalize into the nucleus and orchestrate all this induction of interferon genes. So we chose to concentrate on CXCL10. I did several lines, but I will show you about CXCL10 knockout. So we generate ES cells with that we introduce this knockout cassette into the CXCL10 locus. We're able to pick clones that were differentiated successful into SC-beta. So you can see it by double positive C-peptide or NKX691, which 20% is usually relative good differentiation. You can see the same when you co-stain with C-peptide and glucagon, concentrated on only the monohormonal cells, the ones that have insulin and not other hormones. And you can appreciate, I hope, that when induced by interferon, recombinant interferon in culture, there's almost undetectable level of CXCL10 that is secreted. We see the same in intracellular levels. And to validate this gene hit, we did some more co-culture experiment where we took wild type or CXCL10 knockouts, SC-islets, co-cultured them this time, not only with PBMCs, but also isolated T cells or NK cells. NK cells also have that receptor for chemokines like CXCL10. And with all these co-culture, we measured the rate of apoptosis, and we see that it's being reduced both in general population of PBMCs, but also in T cells and NK cells.
Elad, this immune suppression, or hypoimmunogenicity, is this global? This is not specific, right?
Yes, it's not specific for beta cells. In this instance, I looked at beta cells, but if you looked at alpha cells or there's other EC cells in the differentiation, you see something similar.
Mm-hmm.
Yeah. So we're not saying this is a beta cell response, but we're more interested in protecting the beta cells than the other cells.
Yeah.
So
I think this is an interesting point that Monica mentioned. So you have single cell RNA-seq, so you are allowed, for instance, to do pseudo-bulk analysis on the alpha and the beta cells.
yeah, and again, I wanted to make a short presentation. So if I had time, I would show you the single cell experiment where I did some differential expression between immune challenged SC-beta and control cells. And then I did the same with alpha cells. And you see the same patterns of interferon gamma response genes like HLA. You see CXCL10 in both. I even think it seems like alpha cells even have a bit more response of CXCL10. So it's not a beta cell specific.
But you didn't look at cell death. You are looking at gene expression. I don't know if you listen. I gave a webinar at the HERN, I think 1 or 2 weeks ago, adding single cells, alpha and beta. I think this is available online.
Mm-hmm.
Take a look and perhaps we can discuss later.
Yeah.
Because we see some differences that are subtle, but for instance, alpha cells have much more HLA-E, which is protective. They have more BCL, XL. So even if CXCL10 is the same, as you say, there are some subtle differences that may contribute. Take a look at the webinar and then I don't know if you will be at the ADA meeting next week. No. Well, take a look and then we can, if you find of interest.
Yeah, be happy to talk. Yes.
Via Zoom or whatsoever.
Yeah, it's interesting to see how alpha cells are being able to protect themselves versus beta cells. because they have the same response as we see it, at least in this allogeneic setting.
Hi, Elad. One quick question. Well, maybe two. Right? Did you go deeper into specific CD4, CD8 quantification, and as well as do you see the SOX expression change from your data from this pool of cells?
Yeah. So we do see. We can look specifically in CD8 and CD4. We do see reduction in activation of CD8 and CD4 T cell activation in this co-culture. In terms of SOX1, it does upregulate as part of the whole lighting up of the interferon response. We also did some experiment where we overexpressed SOX1, and it showed promising results too of something similar to what you see here, reduction of the immune response. So it's another nice target to experiment with.
Yeah, I like that approach.
Yeah,
that's pretty cool. Thank you.
Thank you. Just to finish this slides, I did another validation in vivo, which is a longer experiment, but was very important to try, where we took the same HPI model, only now separately transplanted either wild type SC-islets or CXCL10 knockout islets from that line. Parts of the mice were injected with PBMCs and part were left as the controls. And we looked at these yellow cells, yellow bars. These are the wild type, and you can see that with those mice that had PBMCs, insulin level were reduced, while CXCL10 transplanted mice with or without PBMCs, they showed more or less the same level of insulin, and there was no change, implying that they kind of escaped that immune response in that sense, same as we see in vitro. To conclude, we did this broad experiments with CRISPR screen and stem cell and single cell transcriptomics, where we could detect something that was crucial, seemed crucial for determining the fate of SC-islets when they're going to be transplanted. And it seems that interferon response is important to alert the immune system and a strong player in SC-islet immunogenicity. We're able to find potent targets that some of them I couldn't show you all of them today, but we did some experiment with lentivirus to knock out or overexpress specific components of the interferon response, either the receptor or STAT1, either overexpress SOX1, as I just said, and PD-L1. And we considered CXCL10 because it looked very promising. So we went on to generate this line, which seems to have immune evasive properties in both in vitro and in vivo. And it shows that we can lower T cell and NK cell activation by depleting that harmful genes, that harmful chemokine, as we see it. That's it for my slides. If you have more questions, you'd like to discuss more, I think we still have time.
Yeah, we can run a little over because the discussion has been really great. But I wondered if anyone would like to ask another question, just unmute yourself and dive in.
Yeah, again, this is Jean-Luc. So obviously, the CXCL10 deletion is the signaling is downstream of JAK in pathway activation. So what is your thought? Is this very universal of immune activation, i.e., is that the reason why JAK inhibitor being applied in the transplantation world, due to the fact that it mediates the CXCL10 activation? So i.e., what is your opinion of CXCR10 target not only just for the inflammatory potential for autoimmune disease, but in the transplantation world as a universal approach. As to the current JAK inhibitor being applied, but as we know that the side effect is very, very heavy. So what is your opinion of targeting the CXCR12, I mean, CXCR10, I'm sorry, as a alternative to potentially minimize the impact of the side effects?
Yeah, I think I haven't experimented with the JAK inhibitor. It seems like promising to inhibit that immune response. Maybe part of the mechanism is reducing the level of CXCL10, but it could also involve the HLA lowering, reducing HLA presentation. I think it's valuable to reduce the dependency on drugs, using drugs, and if we can avoid that, this is a great way by using a gene-modified line or finding some other ways to reduce it without perturbing too many genes. But yeah, I think we also need to be cautious in depleting the whole interferon response, because if, for instance, you knock out STAT1 or you knock out the interferon receptor, you also lose inhibitory signals like PD-L1 or SOX1. So it's a tricky situation if you silence the whole pathway. I think we need to concentrate on what's most harmful, and we saw that CXCL10 is something that we might want to get rid of. It's not doing really good in our cells, so why do we need it there?
Yeah, can I just ask, if you get rid of the CXCL10, then, what is the risk for sort of a teratoma going undetected, or is there none?
Yeah, so there's always the problem with the risk of making the cells for neoplasma and making cells that are immune evasive to the immune system, because then if there is a tumor, then it will be there and won't be detected. But that will always be a problem even when immunosuppressive drugs or other gene modification that are being targeted nowadays from other labs or companies.
Mm-hmm.
And it's an important issue and needs to be addressed. Finding ways either by thinking really good where you transplant the cells so they can be extracted if needed, or some talk about inducing some suicide genes or other ways to make sure these cells are not harmful in some situation.
Or maybe some kind of new visualization that can really, monitor them carefully and regularly.
Yeah, and there's ways to also get rid as much as possible from those undifferentiated cells that may cause a problem. Well, it's even one cell that still has pluripotency and it's not differentiated, which can cause something that resembles a tumor. You need to get rid of.
Yeah, I wonder if any study out there comparing the CXCR10 knockout versus the PET inhibitor. I mean, JAK inhibitor, I consider that it's more like a broad spectrum inhibition.
Yeah,
you do parallel, right? So you kind of do a JAK inhibitor as comparing side by side with the knockout mouse, and then expose them to pan cancer inducer. See, if just to validate CXCR10 here is that have differentiation compared to JAK inhibitor as a broad effects.
Yeah. So basically, if every perturbation that we do or every line that we do needs to be evaluated in many different ways to make sure it's safe. And there's a whole, I guess there's a whole pipeline. So I just showed one clone, but there should be evaluated like 20 or 100 clones to see that they're the most safe, the most treatable, the best cells that you can use to treat. Right? Right. These cells that I use might be not very good to be transplanted in humans because they don't make enough insulin, for instance. But I just wanted to show the concept that.
Yeah, you're still learning about, what's actually going on in the mice using mice models, and it's all contributing to the knowledge base. Yeah. And I mean, they're very elegant experiments that you've done. And thank you so much for sharing them with us. I did want to ask, like, what's next for you? I mean, are you looking for positions? Are you interested in that? I mean, we're happy to kind of send that out along the wires. Or
yeah, I'm looking for jobs in the industry right now. Yeah.
Fantastic. And you want to continue in this line of work?
I'll try. Yeah. Everything related. There's many interesting things around there. It's not, hopefully I can continue this and this spectrum, but could be other things that might be interesting in the same level of interest.
That's great. I actually have a couple of people in mind that I'm going to set you up with and connect you with after this talk. So I mean, it's beautiful work. Thank you so much.
Great. Thank you
for speaking with us today and showing us this, detailed and elegant work as you're kind of like gently and delicately sort of teasing out these important targets.
Great.
Okay, then.
Thank you so much.
Have a great rest of your day and hope to see you again soon. Thanks again.
Bye.
Thank you.
Bye-bye.